The eDNA Aquatic Kit contains everything you need to collect and preserve eDNA from aquatic environments. In this kit, you'll receive a pair of nitrile sterile gloves, a sampling bag for collecting water, a 60ml syringe, a filter for capturing DNA, preservative solution and filter caps, a specimen safety bag, and a datasheet for noting information about your sample. You will need to take into the field with you a smartphone or GPS for recording location data. Remember to always check and obtain any licenses or permits you need before going into the field. Scan the QR code on the kit bag for full sampling protocol and refer to our guidance on how to choose your sampling locations, how many samples to take, and how to subsample to collect water that is representative of your chosen area. Before sampling, it's important to put on the gloves provided to minimise contamination. Once you have collected your sample of water, you will need to filter it through our enclosed filter to separate out the DNA. Note that the top of the white line on your sampling bag indicates the 5-litre mark. To filter your water, take the large syringe and draw up 50 ml of water. Sometimes air in the syringe can make filtering more difficult, so ensure that any air is removed before attaching the filter. Attach the filter onto the end of the syringe with a small twist, and then push water through the filter. If your water is turbid, this requires a bit of effort. Remove the filter from the syringe and repeat this process until your desired volume has passed through the filter or until the filter is clogged. You will know when the filter is clogged when it becomes too difficult to push any more water through the filter. Measure the water passing through your filter either by filtering into a measuring jug like this one, or continuing to count how many times you've used this syringe. Once you have finished filtering, fill the syringe with air and pass the air through the filter to dry it. Do this 3 to 4 times. Here you will have your eDNA sample. To preserve your sample, take the small syringe from the grip seal bag, uncap it and attach it to the filter via the inlet. Turn it so that the filter is pointing upwards and slowly push the plunger so that the preservative solution fills the filter capsule. When you notice a bubble of solution coming out of the top of the filter, take one of the filter caps and cap the top end. Flip the filter upside down and push the plunger in all the way. It helps to give the filter a little shake to ensure the preservative is evenly distributed. Remove the syringe from the filter and cap the other end. Don't worry if some of the solution foams or spills over. Place your cap filter into the specimen safety bag provided and seal. Fill out the datasheet with information about your individual sample, including a unique sample ID, which needs to be different from any other samples you may have taken. Place both your sample and your datasheet into the kit bag. Your sample is now ready to send to the lab for analysis. Once all of your samples have been collected, you must also fill in the provided electronic sample manifest. To return your samples, we recommend using the cardboard box that your kits were delivered in. As well as your samples, you may also return any plastic waste to us for recycling. Just ensure that it is washed, dried, and sealed in a bag to keep it separate from your samples. Pack out any spare space with paper or packing material. Securely seal the box and post to the address displayed below using tracked postage. If you're sending samples from outside the UK, contact our logistics team who will arrange a courier service for you.